Ladies and gentlemen, welcome to International Workshop Series 2, held by ITD and RC-GERID Universitas Airlangga, entitled Introduction to Conventional Cloning and Mutagenesis PCR and Molecular Biology Application. Before we start, we would like to remind everyone to do the pretest. The link for the pretest will also available in the chat box. Thank you. Welcome. Thank you for your time to attend this session. Before we start the main session, we would like to invite Dr. Laura Namibayamani to deliver her opening speech for this option. So, Dr. Laura, time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Participant offline and also participant online via Zoom. Honorable Professor Maria Indah, as Director of Institute of Tropical Disease, Universitas Erlangga. Honorable Dr. Gandi, our special guest lecturer today, as postgraduate staff in Kobe University, Japan. He also was graduated previously from Kobe University, Japan. And of course, welcome all the participants, offline and online. I have information the participant will be trained in online more than 70 or 70, around 70, but maybe some of them still do not join yet, so we will wait for them. In this opportunity, we would like to say thank you very much for Dr. Gandi to be able as our speaker in International Workshop Series 2 today. This international workshop is held by Institute of Tropical Disease, Universitas Erlangga, in collaboration with Research Center on Global Emerging and Re-emerging Infectious Diseases. Why we call as the International Workshop Series 2? Because previously we also have conducted the same activity, International Workshop Series 1. We have the guest lecture from Professor Kamio Sensei, same affiliation with Dr. Gandi from Kobe University, Japan. If I'm not mistaken, that workshop was held on August 5th when he came to ITD. We know Dr. Gandi are very busy during staying in Indonesia because he only has around 4 days, if I'm not mistaken. He arrived on Thursday, and tomorrow he will leave from Indonesia, go back to Japan. Therefore, we are very happy and appreciate for having you here and to accept our invitation. This is, I think, the second time Dr. Gandi visit to ITD, Universitas Erlangga, because in the first time he ever visit ITD last year. At that time, he also teach us about the laboratory skill, even though he just teach in the limited person in our study group, especially for viral diarrhea study group. And we think that when you will come to ITD again, it is better and more interesting to have a lecture from Dr. Gandi with more participants we invite other ITD staff, and also we offer to participants, others, and this international workshop will be held hybrid, because we have the participant online and also participant in offline. He will deliver the lecture with the topic about the introduction DNA cloning and mutagenesis PCR. This topic is important for us, especially when we are developing vaccine in subunit or making the monoclonal antibody. Later, Dr. Gandi will teach us step by step and sharing his experience using the DNA cloning and mutagenesis PCR in theory, because we have the limited time only today. So maybe in the future we have the other opportunity to have the wet lab, especially for this topic. For the last, thank you very much again for Dr. Gandi for your sparing time, and hopefully you enjoy during staying in Surabaya. And also thank you so much for the participants who joined in international workshop today. Hopefully the lecture can give us the new knowledge and also a new experience and grateful. And keep healthy, everyone. Thank you very much.
Thank you, Dr. Laura, for your speech. Before we move to the main event, I would like to introduce the speaker for today's session, Dr. Rheza and Bawono. Dr. Gandi is a postdoctoral researcher in Kobe University. He finished his medical doctorate in Pemateri University and then did his doctoral study in Kobe University. His research is focusing on in hepatitis. Recently, he published his research about, I'm sorry if I mistake this one, HERC-5 E3 mediates IS-gyrylation of hepatitis B virus. It's proceeding to promote viral replication in Journal of General Virology. So, Dr. Gandi, time is yours.
Good morning. Thank you very much. So, good morning, everyone. Good morning, Professor Mari. Thank you for the invitation to the ITD for the second time. I'm so happy to get to the Indonesia, so especially to Surabaya. So it's very nice city and very nice lab, the ITD, like the home nature. And thank you very much for the Ms. Laura for the opening speech. And today I would like to give some talk about the introduction of the DNA cloning and mutagenesis PCR. So, sorry, just see. This back to the previous slide. Okay, so it's about the cloning. So the first about the cloning. So the clone or cloning is the process of the making identical copies of the organism cell or DNA. So when we talk about the cloning, so as when I was a child or when I was young, so I always imagine the cloning is like in the movie when we clone some of the human or animal. So and previously I think the 1996, so in the about the leadership of the first clone animal, adult animal that is very breakthrough in the and very controversial about that time about the cloning. So this is still also controversial today about the ethical some reasons. And but the point is that the clone is not as that's complicated actually. When we do the making the identical copies, such as the amplified DNA, actually we already done the cloning. So the DNA cloning can be defined as the technique to produce the multiple and identical copies of the DNA fragment of the interest. So in the next, so for the DNA cloning, so DNA cloning is so it can be made with several different approach. The two of the different approaches will be the first by the polymerization reaction of PCR, and also by the cloning of DNA in cells. And in this talk, we will mainly talk about DNA cloning by the PCR. So and then the DNA cloning can be used for various applications, especially for the gene analysis. So we can build the recombinant version of the gene. Also for the new therapy like the gene therapy. So we make the treatment of the certain diseases caused by the gene mutation. So we know that in the human, so some of the diseases is caused by the gene mutation. So for example, the sickle cell anemia. So we mutation in some region, so it makes the hemoglobins less to obtain the oxygen. And then, so we can also make the protein production by the DNA cloning. So we can make the human protein with the biomedical application for benefit. For example, is the mainly like the insulin. So it's very common previously, and it's very life-saving and breakthrough in the science when we can clone the insulin and also the growth hormone. So this next. So we will talk about the DNA cloning steps. So there'll be maybe several different steps in the protocol, but the main thing is this next. So the first important step is we have to prepare the insert and also the vector. So up next, please. So we have the vector. For example, one of the vector was the plasmids. So the plasmid was the small circular DNA molecule found in the bacteria and some other microscopic organisms. So here we can see that the plasmid has several of the components inside of this. For example, for the origin of the replication, so antibiotic resistant gene, and also cytokine marker and the promoter. So here the plasmid also have the special region, so it's called the restriction site, where we can cleavage the restriction site and insert of the gene of our interest. So plasmids are physically separated from the chromosomal DNA and have the origin application. We can see it in this picture is by green one. So this origin of replication allows the plasmid to replicate independently, separate from the replication of the chromosomal DNA. So actually, in the nature, plasmids are present in the some of bacteria. For example, in the E. coli, so the plasmid is already in the nature for a billion years, but we just recently hijacked them to our benefit for the medical engineering or the biochemistry. And also the plasmid can carry various genes. For example, we can put the antibiotic resistant gene. Usually we use like the ampicillin or clindamycin or other antibiotic resistant genes. So we can select the plasmid to which plasmid to be grown, and we can exclude other plasmid that is not inserted by our gene. So next. So about the insert and the factor of the digestion. So actually the first is the digestion of the restriction by the restriction enzyme. So the restriction enzyme is the protein isolated from the bacteria that recognize a specific sequence of the nucleotide and leave the DNA at the specific site. So the restriction enzyme is also used by the restriction nucleases. The process of moving DNA by the restriction enzyme is called the restriction digestion. The plasmid can be digested by more than one different restriction enzyme. And in the cartoon in the right side is the polysaccharide or the restriction enzyme. So usually plasmid has one of the very unique sequence. Just like the we can call it the palindromic sequence. So we can see here in this example is like the GAATEC, and when you read it the backward, it's also can be read the GAATEC. This site is one of the restriction enzyme site, and by the restriction enzyme, this sequence can be cleaved and then can be separated and make the two CPN. So there is blunt end and also the CPN. So in this case, it makes the CPN. So there are so many restriction enzymes, so you can use according to what we need. And usually, this restriction enzyme is in the nature is always the sequence always repeated. So there are a lot of possibility that it can cleave several genes into several pieces. So if we have the one very long gene, so it can be contained of several same restriction enzyme sequence that can be cleaved. So if let's say we have this very long gene and it can be cleaved, I think it's probably around every 4,000 years. So that's why we when using the plasmid, they are usually only contain one of the restriction enzyme sequence, because if we in the plasmid we have very long and multiple, it can be some problem if we cut by the restriction enzyme. Next. So in the restriction enzyme, also we can use the single digestion or the double digestion. So here is the combination about the single digestion and also the double digestion. When we treat the plasmid or the DNA insert, let's say when we use the single digestion, let's say we use the equal one. And also when we use the double digestion, we use like the combination like with the NOT one and EXO one and others combination. So here, when we use the single digestion, so the digested by the one restriction enzyme, of course, and then when we use the double digestion, we use the two different restriction enzymes. So the key is the two different of the restriction enzyme. So when the first thing about the single digestion, it will mix both the same ends as we previously see, both the cut at the same location and both meet the same CPM. But in the double digestion, it will create the different ends because the difference of the restriction enzyme sequence that we cut by the restriction enzyme. So it will make the two different of the end, And then ends in the single digestion will be compatible for each other. So it will, there is a possibility for the self-ligation also. So when we cut the plasmid, so by the restriction enzyme, and then we do the ligation process and we want to ligate with the insert, but there is a possibility of the self-ligation for the plasmid to be ligated again because it has the same end. So it will match and then stick again. So there is a possibility of it. But in the double digestion, so there will not be a compatible ends with each other. So there is no possibility for the self-ligation because we cut in this side and we cut in this other side with the different sequence. So it will not self-ligate. Then in the single digestion, so there is a possibility for the insertion of the foreign DNA fragment in the both orientation. So when we cut also the DNA or gene of our interest, it will also cut in the same ends. So it can be both sides. So the DNA can be this side or the other side. So it can be sticking in the we can say in the proper direction or in the improper direction. So there is a possibility of it because the cut of the section enzyme is the same. But in the double digestion, we can exclude that possibility because of the sticky part that all the will be the ligation part will be the different. So there is no possibility of the improper orientation of the insert. And the plasmid made by the single digestion called potentially contain of the three possibility. One of the self-ligation plasmid without insert, the plasmid with wrong direction or wrong orientation insert, and plasmid with the proper orientation insert. But in the double digestion, we can exclude that, and we can make the plasmid with the double digestion is ensured to be the plasmid with the proper orientation. This next, so we can see here in the this is the scheme of the insert factor cut restriction enzyme. You can see here in the gene of interest on the left side and the bacterial plasmid on the right side, and we can use the restriction enzyme as the function like the fragment. So they will cut or eat the DNA in the certain sequence, and they will make the sticky ends. So these sticky ends next will the next step is the you can see the previously we cut the digestive the insert in the factor, and after we cut the linearized factor it's because the plasmid is the circular, and then we cut by digestion, so it becomes the linearized, so become the linear, and also the tube. So the next step is we need to purify this cut plasmid and also cut the insert. So to do that, we usually use the agar gel. So here we purify the plasmid and also the digestive insert and digestive plasmid into the agar gel. So we load into the agar gel, and after we run the electrophoresis, and we will isolate the digested plasmid and also digested insert and make the gel purification. Next. So this next step is about the insert and the pectoralization. So after we digested, so we cut the plasmid and also the insert, we need to combine both of the plasmid and the gene of the ingress or insert into so we can combine them into one of the plasmid with the inserted gene. And this process is required by the DNA ligase, the ligation process. So this ligation process is very crucial about the step in the cloning. So because without the ligation, so we will not have the cloning with the insert. So here the DNA ligase is a specific type of an enzyme that can ligate or join the ends of the clone DNA fragments. As you can see here in the living organism, DNA ligase also plays essential role in the DNA replication and also in the DNA repair. So this DNA ligase will join or will ligate our sticky end of the plasmid and also the insert gene. So as you can see in the previous cartoon, so in the green one is represent of the previous plasmid, and the red and blue one is the previous insert gene. So here, so about this process, they will join together and make the glue, some kind of like glue, and this bond is very strong, is strengthened by the DNA ligase, and we will have the joint plasmid and the insert gene at the end. Next. So this is the cartoon about the insert and pectoralization by DNA ligase. So here in the left side we have the insert, in the right side we have the plasmid, linearized plasmid. So joined by the DNA ligase at the both ends, and we will have the transport plasmid or the our new plasmid insert. So next. So the ligated plasmid, as we can talk previously by the single digested or double digested. So we can see in the single digested plasmid, there's three possibility of the plasmid. So we can see here in the left, upper left, so with plasmid with the insert. So the direction of the gene is the same as the direction of the other chromosomal plasmid. And then another possibility is the plasmid with the opposite direction or improper direction. So the gene inserted will be in the opposite direction. So this will be another possibility because of the single digestion. And here we in the example of the cutting with the equal one. And also the third possibility is the self-ligated plasmid. So we will have the only plasmid without the inserted gene. So is also a possibility it will happen. But in the double digested plasmid, this possibility can be ruled out to make the plasmid in the correct or the proper orientation. Next. So the next step is we have to transform this plasmid because we only have plasmid in this limited amount of plasmid, and we want to amplify the plasmid. And how to do that is that we have to transform this plasmid into the bacteria, or for example, into the bacteria. This process of transformation. Next. So the transformation is the genetic process in which the foreign DNA is introduced into the host cell. This, for example, the bacteria, and also the yeast, and also the plant cells. And this transformation is typically performed by the chemical transformation or electrophoresis or a particle bombardment. In this chemical transformation, we use the competent cells for example, the E. coli that already engineered to be made competent with the bacterial cell will more permeable. And in the electroporation, we use the electroporation to induce to manipulate for example, bacterial wall to be more permeable. So this using the electric charge. And then the particle bombardment is maybe possibly they use mainly in the plant cell. So we shut the cell with we bombard the cell with our plasmid in the insert, so it will be penetrate the cell. But in this talk, we will mainly talk about the chemical transformation, which is we are using the competent cells in this talk, especially the E. coli. So this bacterial transformation is the process of the gene transfer with some bacteria pick up the foreign DNA from the extracellular environment. So we will introduce our plasmid into the bacterial. So we have to penetrate the plasmid into the bacterial so that it can be replicate itself into the bacterial, while the bacterial also replicate itself. So it will be exponential growth of our plasmid. So the transformation, bacterial transformation was mainly done by chemical transformation and electroporation. So this next. So in this figure, so this is the scheme of the bacterial transformation. As you can see, the first in the yellow one. So we have our plasmid DNA. So we will mix the plasmid DNA with our competent cells, which is in this case the bacteria. And you can see here in the competent cells, they have no plasmid inside of them. So they just the functional DNA. And the next step, after we mix them together, we will incubate them. So it will take some of them to try to mix together. And then when in the next step, we will have to introduce the bacteria with the heat shock. In this case, we will use the temperature of the 42 degrees for about 1 minute. So in this process, we are hoping that some of the bacteria will take our plasmid and pick up our foreign DNA. In this case, it's our plasmid into their inside. With the help of the heat shock, when we heat shock this competent cell, their cell wall, it will be more permeable. So our plasmid DNA is hopefully will easily to get inside of the bacteria. And after that, we have to give the recovery for the bacteria by putting back into the ice, so that we hope that the permeable membrane will be returned to normal again, and that the bacteria will start to replicate itself. And then the next step is the colony-free and the plasmid purification. So next, please. So in this case, after we done the recovery of the transformed cells, then we have to add the nutrient medium. So this is very important because the bacteria will need the medium for them to grow, so they need something to eat. So we add some the nutrient medium into the transformed cell and let them grow. So in this process, we use the medium that not contain the antibiotic of the certain antibiotic, because in our plasmid, for example, we use the plasmid that has resistance to the ampicillin, and in this case, we use the medium without the ampicillin. So we are hoping that all the bacteria will grow together. And the next step, after we incubate them into the 30 37 degrees about 1 or until 3 hours, we hope that there are a lot of the E. coli that already replicate with our insert plasmid or not, it doesn't matter. So for the next step, please. So then we will inoculate of the transformed bacteria into the other plate that contain the antibiotic of our plasmid resistant gene. So if our plasmid contain the resistant gene for the ampicillin, so we have to make the agar with the ampicillin. And if we have the plasmid with the kanamycin, so we have to use the kanamycin plate. So in this process, please do not make sure you have the right right antibiotic plate. So you don't have to undergo this mistake again. So in this step is very important because if the bacteria take up our plasmid, in this bacteria will have our resistant gene of the antibiotic. So after we incubate the agar plate, so after overnight around 16 hours or even 24 hours, we will have the bacteria, growing bacteria that contains our plasmid. So the bacteria that did not take up our plasmid will not grow at all unless we do some of the contamination. But in the ideal settings, the bacteria that contain our plasmid will only grow. So and the next slide is so after that we will pick up this colony of the bacteria and we are growing them into the medium. And we will also growing them in the medium with the antibiotic. So we will make sure that our colony that we are picked up is contain our plasmid. So this is very important. So we have to incubate again and usually overnight. So we have to get from this single colony out, we will have so much of the bacteria that we will need for the next process of the plasmid purification. So about the plasmid purification, we will show in the next video. So later we will see the step by step how do we purify our plasmid. And then after that, we will after we purify our plasmid, it doesn't guarantee that you have the real plasmid because there is a lot of the possibility happen. That's why we have to check the plasmid into the agar shell. So usually because in this conventional cloning, the successful rate is not so high, and for the new technique within the more expensive technique, so they will increase the possibility of the DNA cloning. Like in our lab, we use the like infusion technique. Infusion technique is a lot more expensive, but the step is relatively same, but the successful rate is more than 95%. But in this case, if we done the traditional cloning, the successful rate is not so high. So our plasmid, it can be just the self-ligation plasmid or just with the wrong insert or the proper insert. But by the agar shell, we will distinguish between which plasmid is inserted, contain the insert gene or not, because the time that produced will be different. But by the agar shell, we cannot differentiate between the plasmid with the proper orientation gene or improper orientation gene, because the size will be same, but to do that, we absolutely need the DNA sequence for that. So next is that's called the DNA cloning. So next we will introduce about the mutagenesis. So this the mutagenesis is the mutagenesis is the words from the muta is the mutagenesis. So we make a form of the gene. Muta is gene, genesis is a form. So we will give birth to the gene. So this mutagenesis is the change in the DNA of the organism resulting in the gene mutation. So not all of the DNA mutation, of course, can lead to the change in the protein synthesis or the function. So we always see that the mutation is not always bad. So usually we also always think that mutation is bad. Yes, some of the mutation is bad. So some of the disease is caused by the mutation, but some mutation also can have a non-beneficial effect to their host. Like for example, to the certain viruses. So when the viruses got the mutation, like previously we got the for the recent SARS-CoV-2, the new coronavirus. So they have the mutation on the spike. So it's very concerning about this mutation, whether it's will beneficial for the virus or not beneficial for the virus. But some of the mutation in the southern region of the coronavirus, it has proved to be make this virus is more rapidly infectious, highly infectious, but less severity in the cause, especially in the latest of the strain. But in the beginning of the strain, there will be a very bad strain that is very highly contagious and highly severe. So not all the mutations in the DNA or RNA that makes the change in the protein synthesis or the function. So and also the mutagenesis is one of the important factors for the evolution. So our evolution, a human also evolution of the human also led by the mutagenesis process. So you can see in the people who live in the near the sea and who live in the high altitude, they will have the different percentage of the red blood cell and how many oxygen they can take. So that's also the mutation. So one of the example of the mutation we are talking about here is the point mutation. So the next, so the point mutation is the mutation that occurs within a gene where a single nucleotide base is altered. So this only one single nucleotide base is changed. So here, this point mutation can be a substitution or insertion or deletion. And the point mutation can result in the change of the protein construction and also protein production and protein composition and also the protein function. It depends on the mutation. So here, the example of here is in the top one, the wild type, or let's see the normal one. So here we can see is the amino acids. So here, so we can see this amino acid, and then we have the substitution of the mutation. So we can see it from the AAG. We have a change to the AAG. So from the after only one mutation of this, it's the protein is changed from the lysine into the arginine. So we can see here. And next is the insertion. So when there is insertion of the certain gene, so it is the first in the AEG And then from the AAG, there is an insert of the T, so it becomes the TAA, which is the stop codon. So this will prematurely stop of the become stop codon, and then the protein will entirely change. And then when we a duration of the one nucleotide also will make a change into this phase here. So from the lysine and then previously, and then become the leucine, becomes after one. So from the AAG and then TTT become the AAG and TEG. So it's completely different of the protein construction. So this also can we can see like the parasite mutation. So the next. So the point mutation, the next is can be divided into the several groups regarding of the effect to the functional change in the final protein product. So here, so the first is the silent mutation. So the new codon that code the same amino acid as the wild type. So here, if there is a change like AGA to the AAA, it becomes the same amino acid, but the codon the amino acid is the different. So, but the protein that produced is the same. So we can see here because there's a 20 amino acid, and we see that the one of the protein amino acid has the several codon. So if there is changing that not changing the protein that produced, it's called the silent mutation. And then the next is the Snell sense mutation that result in the stop codon in the position where it was not one before. As you can see in the previous slide, when we add the insertion, it's become suddenly become the stop codon. So it will call the nonsense mutation. And the missense mutation is the substitution mutation that is result in the functionally different amino acid and can be divided into the two groups. So the conservative mutation, like the mutation that create the new amino acid, that is the same type of the original one. As you can see here, there's this is just for the big picture. So we have to distinguish the three different groups, that the electrically charged amino acid, non-polar, and also polar. So here, if we change from the lysine into the arginine, it's called the conservative mutation because it's still the same type of the original. But if we mutate it, the new amino acid is different type. For example, if when the non-polar becomes the polar, or the electric charge becomes the polar, or the polar becomes the electric charge, it's called the non-conservative mutation. So next, please. So this is the table. So just for the diagram for the point mutation. So here in the first in the no mutation. So this is the lysine for example. And then when we choose from the DNA level or mRNA from the AAG to AAA, it's still lysine. So there is no change, but only change one delta, but doesn't change the protein that we produce. So this is called the silent mutation. And the next, when from the AAG become UAG, or from the TTC become ATC, or from the AAG become TAG, it's become the stop codon. So it's called the nonsense mutation because it's become the termination. So there is three, as know that we have the three stop codon. We can make the if we want to remember is UROA, UAA, UR1, UAG, and one UR and UAG, UR1. So UAG, UAA, and UG. And UR1, UG. So UR1 is software. And then for the missense mutation, so we can see in the conservative one and also the non-conservative one. So as I said before, if we change from the lysine, like the AEG become AGG, become the RGN, they are still in the electric charge amino acid, still same type. So it's called the conservative mutation. But when we change into the from AEG to ACG, it's become threonine. It's completely different type of the amino acid. So it's called the non-conservative mutation. I hope you just understand. It's just this type of the threonine mutation. And next, so the Mutagenesis PCR is the molecular genetic engineering and technique for the generating site-directed mutagenesis. And the site-directed mutagenesis is also known as the site-specific mutagenesis and oligonucleotide-directed mutagenesis because only change the one nucleotide. And the site-directed mutagenesis can modify the DNA at the specific nucleotide and change the amino acid and resulting in the protein conformation and protein changes. And the key component from the site-directed mutagenesis are the following. So first is the DNA with the plasmid with insert, and then the target genes nucleotide sequence, and the primer with the modified target. The next please. So in the mutagenesis PCR step, the first is the primer design. This is the very crucial part for the mutagenesis PCR. We have to understand it's very difficult, and we must learn a lot about the primer design. I also still learn much about needs to learn much about the primer design because there's different primer construction, different intention or wants to introduce the related next. So here for the overview for the primer design, the first is the designing primer. It's the critical step of the successful mutagenesis PCR. If we design the wrong primer, it will result in the mismatch or unsuccessful of the mutagenesis. So we will not produce the successful mutant. It will cost us, of course, money and also time consuming because this process is very difficult and very expensive to perform. But when we have the good understanding about how to design a primer, it will be very beneficial for us. So we can make the mutation of what our intended to do. And the next is the primer should not be too long or too short. So ideally it's around 18 to 24 nucleotides. And because if the too short, it will be there will be some incomplete or this failed to the mutation. And if the too long, it will require a longer enzymatic process to do that. And for the TM value, for the ideally it's around 50 to 60 degrees. This is the for the next. If you learn more about the mutagenesis PCR, we can calculate how to do the TM value. So it will also important in the PCR PCR machine. So we have to adjust the TM value. So we will get the correct result, and we will produce the successful mutant. And then the primer should be adjusted according to the kind of intended mutation. So we will have to adjust what kind of the mutation we will introduce, like the substitution, deletion, or insertion. So in the right side, the example. So it's just simple example. So here we use the substitution from the arginine to lysine. So when we this one for the AGG into the AAG, so we only change the G into the A. So we that's why we use the primer, as you can see in the following. So we use in this case we can put the a mutation in the middle of the primer. So for designing a primer, you should avoid the repeating amino acid, because it will cause the maybe some trouble in the next when you do the PCR. So you have to avoid the making the repeating sequence for the plasmid, and it's better to have the GC content in the start and in the end of the primer. And also in the second, we can see the dilation. So we will exclude in the gray area. So GCA we will exclude this one. So in the you can see the next and the before of the dilation of the sequence in the green one. So you can see, and then the green one, when you design a primer, so the green one becomes the close. So you don't have to include the gray one. And the next one, the insertion is the same. So you introduce the new amino acid sequence. So when you design a primer, you add the blue one. So when you chelate, you chelate the gray one. You insert the blue one. So next, please. So in the mutagenesis process, next. So in the mutagenesis PCR, so in the intended mutation will be introduced using the help of the PCR, and then the segmented primer will help to produce the mutated clone apart from the H-parental clone. So they will make the sorry, so I took the wrong picture of that. So, but the PCR cycle for the mutagenesis PCR should be adjusted accordingly. So usually 10 to 15 cycle. If you got too much cycle, you will introduce the possibility of the secondary mutation. So it will not be good for your result. The next is. So the next step is the very crucial part of the mutagenesis PCR. This is the digestion. Next is, so this is the digestion by the DPN1. So in the mutagenesis PCR, we design the primer and then we do the mutagenesis PCR so that we will make the mutagenetic plasmid apart from its parental. So we have the parental, which is wild type, and our mutated plasmid. So in the process, there will be two kind of the plasmid with the mutated gene and the wild type one. And to cleavage to destroy the wild type one, we need the help of the GPN1. So because when we do the mutation, we are making the plasmid is non-methylated, and the parental one is still methylated. So the GPN1 is cleave the methylated plasmid. So only cut the parental, the wild type one, and that's why this. You can see the second GPN1 is the restriction enzyme that targeted the parental line, resulting in the cleavage of the parental plasmid. And the failure or inadequate of the digestion by GPN1 might lead into the unsuccessful mutation. So, for example, if you add the GPN1 and not enough amount, and you incubate the same duration, let's say 3 hours in the 37 degrees, it might not adequate for the GPN1 to treat our non-mutated plasmid, so that our product, when we transport into the E. coli and you repeat the same technique as the DNA cloning. So after this, this is actually the last step of the mutagenesis PCR, because after this we introduce our plasmid after PCR into the E. coli, and we will make the copies and then purify the plasmid and check the sequence. If we are failed in the GPN1, it may lead into the unsuccessful of the plasmid. So I was once forget to add the GPN1, and I got all the clone is the wild type. And then after I add the GPN1 from the 8 of the clone that I took, 3 of them are the mutated plasmid. So you add the GPN1 and the correct amount and the correct incubation period, it will increase your possibility to get the mutated plasmid. Next, please. So after that, the same step for the bacterial transformation. Next, please. So and then in the mutagenesis PCR, the bacterial transformation is principally the same in the DNA coding, and the bacterial transformation in the mutagenesis PCR might require several adjustments, of course, in the amount of DNA you add into the E. coli, and you have to try it several ways also. One of the ways is when you do the previous mutagenesis PCR, you add the different amount of the plasmid. So I was once try the different amount of plasmid, like let's say 50 microgram, then 100, 200, and apparently the 50 doesn't grow, but the 100 grew very well, and the 200 is overgrowing. But when we took the 100, it's for that mutation is very fast, so we have to adjust the amount of the insert DNA, also the GPN1 required. So it will because mutagenesis PCR is very long process. So the problem is that our process is not 100% will successful. So you have to improvise. So you have to by the time you will get the sense of how to do it right, because if you are following the protocol, it might not always right. So you have to improvise, and then that's why it's very challenging, but it's very interesting. So, and then the bacterial transformation in the cross-protein transfer is same mechanism. So we take the foreign DNA, which is our mutated or non-mutated vaccine. So next. So the same ways to do the bacterial transformation. The next, and also the same colony screening way. Next is so same recovery when we incubate the E. coli, and then after that next is so we straight into the agarose gel, and we incubate in the 37 degrees overnight or until 24 hours, okay, depending on your result of the colony. So usually it takes more longer time to grow the bacteria. But I think there is this after we do the plasmid purification, also we have to check to the agarose gel, so make sure that our colony is the inserted gene. So it's contain plasmid and the insert. But in this mutagenesis PCR case, the problem is that you are not always get the mutation version of your interest gene. That's why you need the DNA sequencing. So you have to pick your best clone So to do that is you have to try and error. You should have the sense of which one is the correct colony, which one is not the good one, which one is the most possibly contain the mutation. So not always we have to growing a lot of the colony. Doesn't guarantee that you have the higher possibility of the mutated plasmid. Sometimes when you grow only, you will only have 20 or 25 colony, and then your possibility might be increased. So because of the mutagenesis process. So to confirm that, we need to do the sequence analysis, of course, because it is impossible to just know by the looking at the plasmid same, but we have to confirm again with the sequence. And after that, we check our mutation into the expression. We express it into cells, or you do the qPCR, or you translate it into the cell, and you will also combine with other plasmid, and you can check whatever you want. So if you say you want to check like this virus, this region, and let's say amino acid 100 is important. You think that this region is important, so you will mutate it, this amino acid number 100, and you believe that that this region has the impact on your viral replication. And after you successfully mutated the plasmid and you express into the cells, and then you do the Western blotting, or you do the qPCR, and you have the proof that, oh, it's really important, then you will have the good result to be published in the paper. So you see that the most of the published, I think the good paper, they always introduce with the this protein is targeted by this area, or this protein is this domain is very important, especially this region. So because we compare with the wild type one, there is a difference. Or if you use the mice or the animal, so you introduce the mutation, and this mutation actually make the mice like die younger, or they have the disability in their one of the limb or missing or something like that. So by the mutation, you introduce the mutation, your possibility for get the very high quality paper will be increased. So that's why the mutagenesis PCR is very important. So if you master it, so you can make a lot of combination. So not only you will introduce one mutation, you can make the two mutations, three mutations, and for example, in our labs, so we use we mutated the lysine into the arginine. So all of the lysine residue in the certain area, we will mutate it into the arginine. And when we get the lysine which is it doesn't contain the lysine amino acid, we saw that this replication is affected, so that we can compute that this mutation is very have a high impact on the replication of the virus. So, for example, it's just like that. So, in that case, you have to make a lot of the mutation. So, and also you can combine with like if you in your case like the norovirus, coronavirus, you can cut the amino acid. Like say, if the one virus has the 500 amino acid or 150 amino acid, you can cut into the from the 1 until 100 or 150, or from 50 to 100. So you can compare which region is important for like virus replication or virus entry. So you can always make not only the deletion, also you can insert some of the gene. So that's all of the possibilities for the mutagenesis PCR and the application. So this next. So for the importance, as you can see here, so the mutagenesis PCR can help to gain better understanding about the role of the certain protein function and the role of the region of the gene, of course, the effect of the point mutation on the protein structure and the function, etc. So the mutagenesis PCR also contribute to the development of the gene-targeted treatment, vaccine development, of course, and other molecular treatment that will be beneficiary for us. So you can develop the vaccine for certain viruses by the mutagenesis PCR in the base of your project. So and then the mutagenesis PCR also have the great impact on the studies of the pathogen. So when you have the, like we say, the SARS-CoV-2 virus, when the mutation in this region, and then it's become more infectious or more rapidly. So and then you can compare. So like you can get the data from the sequence, like the coronavirus sequence in the worldwide, when this what the kind of the strain is now in dominance. So you can check their sequence and then you compare with the wild type sequence. Which one, like in the nuclear capsid region of the SARS-CoV-2, there's a change in the amino acid. Let's say number 203 and 204, for example, or the P61. For example, so that's why which amino acid is different from the wild type, and what's the impact of this this mutation, whether it will be beneficiary for the virus or not beneficiary for the virus, because mutation not always a beneficiary for the virus or the other proteins. Okay, I think that's all. Thank you very much. So if you have some questions, please.
The discussion session will be held after video. Thank you. So thank you, Dr. Gandhi, for interesting lecture about the conventional cloning and mutagenesis PCR. After this, we will watch the web lab simulation. The video is made by Dr. Gandhi himself.
Baik, saat ini kita mau melakukan untuk restriction enzyme atau digesti dari vektor dan CD.
So, previously I want to apologize that I made this video with the Indonesian language. So, probably if you have to want to in the English version, we can get the translation for that. So, this you can ask whether you want to ask about which step is this or which one.
Baik, saat ini kita mau melakukan untuk restriction enzyme atau digesti dari vektor dan cDNA. Di saat ini kita siapkan vektor, kemudian cDNA, kemudian kita juga siapkan buffer untuk restriction enzimenya, kemudian juga restriction enzimenya saat ini kita pakai not one. Kemudian kita siapkan 2 job, satu untuk vektor, satu untuk cDNA, dan kita perlu DW untuk dilusi. Baik, kita pertama kali masukkan DW dulu. Selanjutnya kita akan masukkan vektor dan cDNA. Pertama kali kita masukkan vektor. Kemudian kita ambil cDNA. Kemudian selanjutnya buffer Setelah itu kita masukkan restriction enzyme-nya. Selanjutnya kita akan inkubasi di suhu 37 derajat selama 3 jam di mesin PCR. Baik, kita siapkan mesin PCR-nya. Suhunya 37 derajat selama 3 jam. Ambil bertanya, ini masukkan dalam mesin PCR. Dan kita bisa mulai. Kita akan lanjutkan lagi setelah 3 jam inkubasi. Baik, setelah 3 jam kita akan ambil, kemudian kita akan lanjutkan untuk proses gel purification untuk mendapatkan vektor dan si DNA yang sudah purified. Sekarang saatnya kita untuk purifikasi dari vektor yang sudah kita digest tadi, yang kita sudah potong vektor dan si DNA yang tadi dari mesin PCR. Setelah 3 jam di suhu 37 derajat, sekarang saatnya kita purifikasi di medium agarose gel. Kita akan lakukan elektroforasi. Yang kita perlukan pertama adalah agarose gel. Kita masukkan di mesin elektroforesis. Oke. Kemudian selanjutnya kita akan butuh loading buffer. Kemudian kita juga akan butuh DNA letternya. Ya, kita tunggu beberapa saat agar dia turun ke bawah dahulu. Kemudian kita akan mulai elektroforesis. Prosesnya kira-kira selama 30 menit. Ya, nanti kita akan kembali lagi 30 menit lagi. Baik, sekitar setelah 30 menit kita matikan. Kita cek, ya, marker biru sudah ada di bawah. Sekarang kita siap untuk memotong gelnya. Ya, sekarang kita akan coba untuk potong gelnya. Kita sudah siapkan 2 cup untuk vektor dan cDNA. Juga kita siapkan mesh untuk memotong gelnya. Juga lampu UV. Kita akan coba potong di bawah sinar UV. Ya, di sini kita lihat vektor cukup jelas terlihat, dan kita akan potong gelnya. Kemudian kita juga akan potong si DNA-nya. Kalau di sini sepertinya tidak kelihatan di kamera karena sangat kecil. Kira-kira untuk gel cutting akan seperti itu. Selanjutnya nanti kita akan coba untuk purifikasi gel dengan purification gel tip. Baik, setelah kita tadi potong gelnya, gelnya masih berbentuk gel. Kemudian step selanjutnya adalah kita akan melarutkan gel ini agar bisa untuk kita filtrasi. Nantinya akan kita masukkan ke dalam collection tube dan filtration tube ini dengan bantuan dari membrane binding solution. Ya, pertama kali kita akan tambahkan membrane binding solution ke dalam gel ya. Kita lihat sudah bercampur dengan gelnya. Selanjutnya akan kita sedikit vortex dari gelnya agar mudah untuk bercampur. Kemudian kita akan inkubasi di suhu 60 derajat selama kurang lebih 3 menit. Kita akan inkubasi di suhu 60 derajat. Di sini kita masih bisa melihat gelnya ini masih ada di dalam larutan tadi. Kemudian kita akan inkubasi selama kurang lebih 3 menit. Kita harapkan gel sudah akan larut setelah 3 menit. Ya, setelah kita tadi lumerkan gelnya, sekarang kita sudah mendapatkan larutan. Saatnya kita untuk melakukan penyaringan ke dalam filtration tube. Kita masukkan seluruh larutan gel tadi sini. Kemudian selanjutnya akan kita centrifuge dengan full speed selama kurang lebih 1 menit. Ya, ini kita sudah saring. Kemudian langkah selanjutnya adalah kita akan tambahkan membran wash solution atau washing buffer ya. Kita akan lakukan washing sebanyak 2 kali siklus. Selanjutnya kita centrifuge kembali dengan kecepatan penuh selama 1 menit. Ya, kita sudah melakukan washing tadi. Setelah kita centrifuge, kita akan lihat sisanya bahwa washing buffernya turun ke bawah. Selanjutnya kita akan washing kembali, tapi sebelumnya kita akan buang dahulu. Baik, kita akan washing untuk kedua kalinya. Ya, setelah washing yang kedua, saatnya kita lanjutkan untuk sentrifugasi dengan dry centrifugation. Jadi kita tanpa menambahkan apapun. Kemudian sebelumnya kita buang dulu terlebih dahulu. Ini washing buffernya. Baik, saatnya kita lanjutkan dengan empty centrifugation. Setelah empty centrifugation, kita sudah mengeringkan dari filtration tube dan semua sisa-sisa residu sudah ada di dasar dari collection tube. Selanjutnya kita akan melakukan elusi. Di sini kita akan menambahkan DW. Kemudian kita sudah siapkan 2 tube yang kita sudah beri nama sebelumnya. Kemudian kita selanjutnya akan centrifuge dengan kecepatan penuh selama 1 menit. Ya, di sini kita sudah melihat vektor dan insert yang kita purifikasi sudah kita dapatkan di dasar tube ini, dan kemudian kita akan simpan dan lanjutkan untuk step selanjutnya, yaitu ligasi dari vektor dan insert kita. Baik, sekarang kita masuk ke proses untuk ligasi. Kita tadi sudah purifikasi vektor dan juga purifikasi cDNA dari gel. Kemudian kita saat ini perlu sebuah ligation mix. Oke. Kemudian kita masukkan ke dalam tube PCR. Dan kita tutup, dan kita siap untuk proses ligasi di di mesin PCR dengan suhu 16 derajat. Untuk ligation mix ini kita hanya perlu waktu sekitar 30 menit saja. Baik, kita mulai untuk proses ligasinya. Kita ambil sampel kita masukkan sampelnya. Sudah kita atur sebelumnya 16 derajat selama 30 menit. Ya, nanti kita akan tunggu bila sudah selesai. Baik, selanjutnya kita akan melakukan transformasi bakteri. Di sini kita siapkan tabung kosong. Kemudian yang paling penting adalah kita butuh kompeten sel. Di sini kita memakai kompeten sel ECOS DH5 alpha. Ini E. coli. Ini kita ambil E. coli. Kemudian kita ambil dari provinsi ini sekitar dari kemudian kita akan diamkan selama kurang lebih 5 menit sampai 30 menit. Setelah itu kita akan melakukan heat shock untuk agar plasmid yang kita buat bisa masuk ke membran dari E. coli. Baik, setelah kita lakukan diamkan tadi selama kurang lebih 5 menit, selanjutnya kita akan lakukan heat shock dengan suhu 42 derajat selama 1 menit. Oke, sekarang kita akan coba masukkan. Kemudian kita start. Baik, kita akan siap untuk ambil E. coli-nya. Ya, kita masukkan. Baik, setelah proses recovery tadi, sekarang kita akan lakukan penambahan media untuk tumbuh kembang dari E. coli kita tadi. Di sini kita melakukannya dengan menggunakan LB medium. Kemudian kita tinggal tambahkan ke dalam larutan yang E. coli yang sudah kita transformasi tadi. Selanjutnya kita tinggal inkubasi di suhu 37 derajat dalam shaker 37 derajat selama kurang lebih 3 jam. Ya. E. coli yang tadi kita sudah campurkan dengan LB medium, saatnya kita inkubasi di shaker selama dengan suhu 37 derajat selama kurang lebih 3 jam. Baik, kita sudah sampai untuk step agar strict. Tadi kita sudah siapkan plasmid yang kita sudah transformasi ke dalam E. coli, kemudian sudah kita inkubasi, dan sekarang bakteri E. coli-nya sudah mulai banyak berkembang dan ditunjukkan dengan warna keruh. Kemudian kita siap untuk menghapuskan ini ke dalam LB plate yang sudah kita siapkan sebelumnya. Dan LB plate ini mengandung ampicillin sehingga plasmid kita yang tadi resisten dengan ampicillin diharapkan bisa tumbuh, sedangkan bakteri E. coli yang tidak tertransfeksi atau tertransformasi oleh plasmid tidak akan tumbuh. Ini adalah kandungannya. Oke, kita lihat. Biar siap semua terus. Ambil dari transformasi. Biar langsung nggak terus. Sama-sama. Bila sudah dirasa cukup, kita lanjutkan untuk step berikutnya. Kita akan inkubasi LB agar plate ini di suhu 37 derajat Ini LP agar yang tadi kita siapkan untuk hapusan. Saatnya kita masukkan ke dalam inkubator. Jadi kita tunggu hasilnya untuk besok pagi. Ya, saatnya kita lihat koloni yang kemarin kita masukkan. Dari sini kita bisa lihat tampak ada beberapa koloni yang bisa kita ambil. Baiklah, kita lanjut ke step berikutnya. Oke, step pertama kita lakukan penomoran terlebih dahulu. Di sini kita menggunakan medium yang sudah memiliki antibiotik di dalamnya. Kebetulan saat ini kita menggunakan ampicillin karena di dalam plasmid yang kita ligasi kemarin mengandung gene resisten untuk ampicillin. Kemudian kita siapkan. Kita siapkan untuk mediumnya. Kita ambil beberapa mili. Selanjutnya kita untuk pick up koloni. Kita coba ambil koloni yang paling baik. Baik, pengambilan koloni sudah selesai. Nah, kita akan melanjutkan untuk inkubasi di shaker suhu 37 derajat. Kita masukkan ke dalam shaker. Ya, kita akan ambil koloni yang kita kemarin tumbuhkan. Ya, kita cek sudah keruh ya semuanya, menandakan sudah E. coli-nya sudah tumbuh dan siap kita untuk purifikasi plasmidnya. Ya, kali ini kita mau purifikasi sel yang kita tumbuhkan kemarin, dan kita akan melakukan purifikasi terlebih dahulu. Kita akan membuat pelet daripada E. coli ini. Kemudian kita centrifuge. Di sini kita bisa lihat ada pelet dari E. coli ini. Kita akan ulangi lagi hingga semua koloni E. coli tersentrifuge. Ya, sekarang kita buang sepenuhnya dan menyisakan hanya pelet. Kemudian kita masukkan sisa larutannya. Ulangi juga untuk semua sampel yang lainnya. Baik, setelah kita selesai sentrifugasi, kita mendapatkan pelet dari E. coli. Jadi tujuan dari sentrifugasi tadi adalah memisahkan sel atau E. coli-nya dengan medium tumbuh E. coli sebelumnya. Kemudian kita lanjut ke step berikutnya untuk melakukan resuspend dari pellet E. coli-nya. Di sini kita menggunakan resuspense buffer. Tujuannya adalah untuk melarutkan kembali E. coli-nya. Bisa kita lihat di sini E. coli-nya sudah resuspense. Kemudian selanjutnya kita akan menggunakan lysis buffer. Tujuannya agar untuk memecah lipid sel sehingga produk-produk di dalam selnya bisa keluar dari sel. Kita akan goyangkan sampai dia lysis ditandai dengan warnanya yang jernih. Bisa kita lihat sekarang warnanya jadi jernih. Kemudian step terakhir adalah kita lakukan lagi dengan neutralization buffer. Jadi ini gunanya agar plasmid bisa renature lagi dan menyingkirkan kemungkinan dari kontaminasi kromosomal DNA, karena kromosomal DNA terlalu panjang sehingga tidak bisa renature hanya dengan neutralization buffer. Kemudian kita shake kuat hingga berubah menjadi warna putih. Next, selanjutnya adalah kita centrifuge kembali selama kurang lebih 15 menit untuk memisahkan isi dalam larutan antara yang soluble dan non soluble. Kita akan mengambil larutan solublenya yang dimana plasmid juga ada di sana. Nanti kita akan ambil supernatannya. Kita akan centrifuge selama 15 menit dengan kecepatan penuh. Ya, setelah kita tadi centrifuge, kita sekarang bisa lihat ya di sini supernatanya terpisah, dimana yang ada di supernatan adalah yang soluble termasuk adanya plasmid. Kemudian kita selanjutnya akan memindahkan ini ke dalam filter tube dan juga collection tube untuk disaring dari plasmidnya. Selanjutnya kita akan buang larutan yang ada di collection tube ini. Kemudian kita akan tambahkan dengan volume wash solution atau washing buffer. Di sini mengandung etanol, di mana gunanya untuk memisahkan plasmid dari materi kimia lainnya, karena plasmid akan terpresitivitasi dengan adanya etanol. Nanti selanjutnya kita akan centrifuge lagi selama 1 menit. Baik, setelah itu kita akan lihat washing buffer ada di bagian bawah. Kita akan membuang washing buffernya. Oke, step selanjutnya adalah kita akan centrifuge kembali. Kali ini kita namakan dry centrifuge, karena kita tidak akan menambahkan apa-apa. Tujuannya adalah untuk menghilangkan washing buffer atau etanol yang masih tersisa, agar tidak mengganggu dalam proses selanjutnya. Setelah kita melakukan dry centrifugation, kita lihat masih ada sisa-sisa etanol yang tersisa. Kemudian kita sudah biarkan sejenak untuk memberikan kesempatan agar sisa-sisa etanol yang masih ada untuk menguap. Kemudian selanjutnya kita akan melakukan elusi. Kita akan mendilusikan DNA atau plasmid yang sudah kita saring tadi, yang terpresipitasi dengan etanol. Kita akan larutkan lagi dan kita akan simpan di tube. Di sini kita cukup hanya menambahkan dengan pelarut, menggunakan beberapa pelarut, tapi kita di sini menggunakan DW. kita akan menggunakan nucleus free water. Kita akan ambil beberapa, kemudian kita masukkan di tengah-tengah persis. Kemudian setelah itu kita akan centrifuge kembali selama 1 menit, dan kita akan mendapatkan plasmid yang sudah terdilisi di collection tube kita nantinya. Baik, kita sudah mendapatkan plasmid yang sudah terfiltrasi. Untuk step selanjutnya kita akan cek plasmid kita yang kita buat di dalam agar-agar gel, karena tidak dalam proses DNA cloning konvensional ini tidak menjamin 100% bahwa yang kita dapatkan adalah plasmid yang ada insertnya. Ya, kita sudah sampai tahap untuk melakukan gel electrophoresis. Kita akan cek plasmid yang kita bikin tadi. Kita sudah siapkan 4 tube karena sampel kita ada di 4. Kemudian kita pertama kali adalah memberikan DNA letternya dulu. Kemudian kita masukkan sampel satu persatu. Ya, kita akan lakukan electrophoresis, dan kita akan lakukan. Nanti kita tinggal menunggu hasilnya. Ya, ini dari hasil electrophoresis tadi, kita bisa lihat di sini plasmid nomor 1, 2, dan 4 ternyata tidak ada insertnya, dan plasmid nomor 3 kita dapatkan insert. Jadi nanti kita tinggal kembangkan, kita amplify lagi plasmid nomor 3 supaya kita mendapatkan jumlah plasmid yang cukup untuk eksperimen selanjutnya. Bisa kita lihat di sini juga gelnya. Oke. Ya, jadi untuk proses cloning ini sudah tahap final karena kita sudah mendapatkan satu koloni yang berhasil kita cloning. Ya. Baik, saat ini kita baik, sekarang kita mencoba untuk melakukan mutagenesis PCR. Di sini dalam kasus ini kita akan coba ubah salah satu nucleotide dari plasmid wild type yang akan kita rubah dengan bantuan F primer dan reverse primer. Dan juga kita di sini butuh buffer. G-Flex, di sini kita memakai produk G-Flex PCR buffer yang isinya adalah Mg2+, kemudian DNTP juga. Kemudian di sini kita perlu juga CKS G-Flex yang isinya adalah DNA polimerase. Di sini kita juga butuh DW. Saatnya kita mulai. Kita masukkan DW terlebih dahulu. Kemudian selanjutnya kita masukkan forward primer. Kemudian kita masukkan reverse primer. DNA-nya, plasmidnya wild type. Kita masukkan. Selanjutnya kita akan masukkan TKS G-Flex buffer ini. Kita ambil, tutup, kemudian di job ini kita masukkan. Jangan lupa kita untuk lakukan mix. Kemudian terakhir kita masukkan polimerase dan tidak perlu untuk dilakukan pipetting. Dan sudah selesai, jadi kita tinggal lakukan PCR dengan menggunakan mesin PCR. Baik, ini setting untuk mesin PCR-nya dalam kondisi ini. Kita akan masukkan zat yang kita larutkan. Kemudian kita akan melakukan mutagenesis PCR. Yang akan memakan waktu selama kurang lebih 2,5 jam. Kita start. Baik, setelah tadi kita PCR untuk mutagenesis, sekarang kita masuk untuk step paling penting dalam mutagenesis PCR, yaitu kita akan mendigesti dengan DPN1. Kita memakai DPN1 karena dia akan mendigesti dari plasmid yang mengandung methylate. Dan dalam kasus ini adalah plasmid dari parental strain-nya, yaitu dengan plasmid wild type-nya. Kita akan ambil sedikit, kemudian kita masukkan dalam PCR mix-nya. Kemudian kita akan inkubasi selama 3 jam di suhu 37 derajat Celcius. Kita akan mulai. Nanti setelah 3 jam selesai, kita akan melanjutkan dalam proses transformasi. Kemudian kita akan tumbuhkan di agar plate. Baik, ini setting untuk mesin PCR-nya dalam kondisi ini.
And also the Mutagenesis PCR. You have time?
We still have time for 2 questions. Maybe you can directly ask and will be directly answered by Dr. Gandi. Anyone? For participant in Zoom, you can leave your question in the chat box. No question? No question for the Zoom participants. So we'll leave the question here. Anyone have questions?
Good afternoon. Good afternoon.
Good afternoon.
I don't know. Can I speak in bahasa?
Of course.
Ya, terima kasih. Ya, ini adalah sesuatu hal yang baru, terutama yang mutagenesis PCR ya. Ya, untuk cloning mungkin saya waktu pernah pelajarinya, tapi karena jarang sekali memakai, jadi agak lupa reminder. Untuk yang mutagenesis PCR, saya masih belum bisa membayangkan. Jadi proses PCR itu sendiri apakah sama seperti konvensional PCR untuk mengaplikasi? Karena kok saya lihat lain-lain kok seperti cloning gitu ya.
Yeah. Okay. So I answer in Indonesian. In Indonesia. Untuk proses mutagenesis PCR yang membedakan hanyalah ketika kita membuat mixture campuran dalam kita akan perlu primer, forward primer, reverse primer, kemudian kita perlu buffer yang kemudian isinya nucleotide, kemudian isinya yang lain tergantung dari kitnya ya. Sebenarnya kasus ini kita pakai simplex, takaran simplex, kemudian ada polimerase juga. Dan yang komponen yang penting hanyalah plasmid yang kita mau ubah, kemudian bisa forward primer dan reverse primer. Sebenarnya intinya itu sama dengan DNA cloning, hanya di sini kita menggunakan bantuan primer itu untuk ketika kita proses PCR, kita ada link ketika DNA kita pecahkan dari glucose, kita pisahkan, kemudian kita tempelkan dengan primer. Kemudian kita harapkan ketika proses selanjutnya dalam PCR primernya mulai dengan bantuan nucleotide yang mulai membuat dari template-nya. Nah, dari kita membuat template-nya itu sendiri, karena kita membuat seksi yang tertentu ya, karena tadi kan bikin nucleotide hanya 24 per forward primer yang mana itu saling overlap. Jadi ketika kita ubah satu nucleotide, maka primer itu akan nempel, kemudian dia akan dengan bantuan kit kita, dia akan membuat mutasi. Tentunya tetap ada yang wild type ya, yang dia tetap masih normal. Dan, maka dalam satu proses PCR itu akan terjadi dua proses, penggandaan yang masih wild type, kemudian penggandaan yang kita sudah jadikan instan. Bedanya adalah di sini ketika kita mendesain primer dengan mutasi itu, kita mengubah dari plasmid wild type kita metilennya, dia ada khusus metilennya. Ketika kita ubah dalam mutagenesis PCR, plasmid yang mutasi tidak mengandung metilen. Jadi perbedaannya di situ. Itulah kenapa kita menggunakan bantuan digestation enzyme DPN1 tadi. DPN1 itu secara khusus yaitu mentarget plasmid yang mengandung metilen. Jadi nanti mungkin bisa dibaca tentang bedanya methylated dan methylated. Ketika plasmid methylated ya dalam kasus ini yang wild type, dia akan dipotong, dipecah. Jadi ketika plasmid itu di-cleavage, pecah, rusak, akan diharapkan yang tersisa adalah yang mutan. Itu hanya saya mutan. Tapi itu secara teori, tapi secara kenyataannya tidak semua yang wild type akan terpecah, karena dalam proses mutagenesis juga akan dari primernya itu juga bisa jadi dia gagal mengubah sekuensinya dan tetap menjadi wild type yang mana juga akan jadi probabilitas. Jadi untuk membuat mutan itu terlalu susah, bukan terlalu susah gampang. Jadi ya kadang kita butuh, kalau andalannya gede bisa dicoba. Jadi kalau proses mutagenesis itu lebih banyak artnya gitu. Jadi kita mengutak-atik, ternyata kalau kita pakai template DNA yang buatannya tadi pada proses kita campur dengan forward primernya, tentunya dengan kalau dari protokol kan forward primer, kemudian buffer dan polimerase itu relatif konstan ya variabelnya. Karena kita hanya bisa membuat berapa jumlah konsentrasi dari plasmid yang kita masukkan. Kadang itu semakin sedikit yang kita masukkan, itu kadang tingkat keberhasilan menjadi mutanya malah lebih besar. Tapi bisa juga ketika kita menambahkan yang lebih banyak, kemungkinan juga bisa lebih besar. Itu tergantung dari plasmidnya sendiri. Karena plasmid kan juga plasmid kan dari plasmid yang misalkan kita bikin plasmid rotavirus atau sapovirus atau norovirus. Di bagian norovirus salah satu gennya saja. Kalau kita bikin dulu kan terlalu banyak. Misalkan dari bagiannya apa yang biasanya kapsid gitu ya, kapsid misalkan itu ya dari gen itu kita cuma mau mengubah mengubah satu atau mungkin kalau mau bikin beberapa mutasi juga bisa ya. Tentunya nanti berpengaruh pada desain primernya. Kalau mutasinya itu sebelahan bisa kita coba bikin langsung sebelahan dua mutasi sebelumnya, atau kita bikin satu primer untuk memutasi satu nucleocapsid, kemudian kita desain satu primer lagi di mana isinya langsung dua mutasi. Jadi kita step by step atau bisa begini. Kemudian dari plasmid itu sendiri kan beda-beda. Kita ada tagnya. Kita mau tambahkan tag, plata atau tag atau tag yang lainnya. Kemudian promoter dari plasmid itu juga berbeda. Kita mau pakai PCMP, PCAG, CDF1, juga tipikalnya berbeda-beda. Jadi kita seringnya di situ. Nanti kita harus pelajari bahwa sama-sama misalkan nukleokapsid, tapi yang satu tagnya beda, yang berbeda. Bisa jadi ketika campurannya mirip, hasilnya sudah beda. Kadang kalau misalkan kita bikin tag A itu lebih gampang daripada tag B, itu bisa juga terjadi. Jadi lebih tagnya itu. Jadi memang prosesnya adalah dengan bantuan PCR dan mirip dengan DNA cloning, karena pada prinsipnya kita akan menggandakan plasmidnya itu. Hanya bedanya kita menggunakan restriction enzyme untuk membantu kita menyingkirkan yang tidak mutan. Jadi di PMR hanya memotong yang methylated itu, sehingga diharapkan ketika kita ekstrak tadi itu, tetap semua wild type ataupun mutan tetap akan tumbuh karena dua-duanya mengandung antibiotik resisten yang sama. Dan kita dari kelihatan pun tidak akan secara umum tidak akan terlihat beda. Cuman satu-satunya cara adalah kita lakukan purifikasi dan kita sequence, itu adalah yang memang sama dengan cloning pada ini, karena kita juga membutuhkan penggandakan. Cloning kan tujuannya untuk menggandakan. Hanya dengan suhu TM value yang berbeda, kemudian lamanya beberapa penyesuaian adjustment. Tadi ada salah satu suhu kita memakai selama 3 menit karena DNA yang kita pakai itu size-nya terlalu besar. Kalau DNA-nya lebih size-nya kecil, kita bisa kurangi waktunya 1 menit, 2 menit. Jadi banyak variabel yang akan kita putar di situ, tapi intinya memang prosesnya sama dengan DNA kronik, hanya dengan bantuan yang berbeda, tapi masinerinya sama.
Itu optimasi juga ya.
Iya, betul optimasi, tapi biasanya sih yang mirip-mirip. Dan juga ada maksudnya kalau kita sudah melakukan plasmid sebelumnya lebih. Tapi untuk mencapai optimasi itu yang paling memakan waktu.
Mungkin saya bertanya ya, melanjutkan apa yang tadi ditanyakan oleh Mbak Dinar. Ini dalam bahasa Indonesia aja ya. Ya, terima kasih Mas Gandi atas presentasinya, luar biasa. Tadi detail, komprehensif terkait dengan step-step bagaimana kita melakukan DNA cloning, kemudian juga mengarah ke satu metode yang disebut sebagai mutagenesis PCR. Dan tadi yang dicontohkan Mas Gandi ini terkait dengan site-directed mutagenesis. Sebenarnya juga kan ada metode yang lain ya, seperti error prone PCR. Itu juga sebetulnya tujuan akhirnya adalah kita mendapatkan suatu mutasi yang itu by targeted. Jadi target kita apa dan yang itu nantinya kita lakukan screening seperti itu di ya. Dan pengalaman memang cloning itu tidak mudah. Jadi kalau dari penjelasan tadi Mas Gandi itu sepertinya mudah ya, tinggal cemplung gitu kan. Harapannya langsung satu step itu sudah hasil produk yang kita inginkan itu tercapai ya seperti itu. Tapi mungkin pengalamannya Mas Gandi bertahun-tahun ya atau minimal 1 tahun ya Mas Gandi untuk mendapatkan satu clone itu. Nah ini mungkin pertanyaan dari saya tadi menyambung apa yang disampaikan oleh Mbak Dinar. Memang menarik untuk melakukan proses site-directed mutagenesis. Cuman gini, mungkin ada trik atau ada saran dari Mas Gandi, bagaimana kita kita kan harus tahu dulu ya, kita mapping kira-kira target mutasi apa yang kemudian kita rubah. Artinya yang tadinya itu wildcard, tapi dengan sengaja kita merubah sehingga karakteristik dari mungkin seperti mikroorganismenya itu kan sesuai dengan harapan.
Ya,
jadi memiliki sifat yang berubah, tidak seperti wildcard. Nah, karena ini adalah site-directed mutagenesis, berarti kan yang kita otak-atik adalah primer. Entah itu forward, entah itu reverse. Jadi kita pilih misalkan salah satu mutasi di bagian 2 primer itu. Nah, selama ini pengalaman itu kalau kita melakukan amplifikasi dengan primer, itu kan produk ujung forward dengan reverse kadang kan tidak bagus ya, artinya kan ada proses kerusakan di ujung-ujungnya itu.
Betul.
Nah, ini bagaimana kemudian saran dari Mas Gandi, mungkin ada pengalaman titik mutasi yang kita ambil itu apa? Apakah dari yang lebih baik ya, apakah dia menjauh dari ujung 5M-nya, atau misalkan kalau kita ambil yang dari reverse berarti kan dia menjauhi dari 3M ya. Nah, ini mohon saran dari Mas Gandi.
Saya coba menjawab ya. Memang untuk pendesain primer itu kan ya susah-susah gampang ya, tergantung kalau ya sequence-nya ada yang gimana gitu ya. Jadi kan kita sebenarnya sih ada, kalau kita lihat dari cara mendesain primer itu ada beberapa guidance ya, kita bisa baca nanti. Biasanya sih dari kalau kita searching di gym itu ya ada salah satu panduan untuk primer design. Dia akan memperhitungkan apa sih yang akan kita perhitungkan. Beberapa yang harus kita perhitungkan itu adalah yang pertama jelas kita posisi mutasinya di mana. Itu kan penting ya. Akan mau kita di type N atau 3N ya, kan? Kemudian kita juga harus mapping dari sekitaran itu mutasinya itu sekuensinya bagaimana. Apakah ada nggak yang pengulangan atau adakah yang tidak pengulangan, atau apakah cukup biasanya sih kalau untuk mendesain primer mutasi kita perlu GC-nya itu kandungannya sekitar 40-60%. Jadi kalau sekitarannya itu tidak terlalu banyak GC kombinasinya, kita bisa pertimbangkan nanti posisinya agak berubah di mana gitu. Jadi untuk forward primernya, untuk reverse primernya juga. Kemudian juga kita harus perhatikan juga amino asid apa sih yang kita mau ubah situ. Yang itu penting untuk desain primernya. Sebisa mungkin sih kita hanya mengganti satu nucleotide, karena kalau misalkan kita dari AAG menjadi AGG kan lebih mudah daripada AAA menjadi AGG. Contohnya seperti itu, karena kan kita harus mengubah nucleotide. Jadi sebisa mungkin kita harus ubah sebenarnya terserah mau kita ubah menjadi sama yang type-nya sama, misalnya sama-sama polar atau sama-sama non-polar, atau berubah dari non-polar menjadi electric charge, itu tergantung dari salah satunya tergantung dari tujuan kita untuk apa. Kalau misalkan kalau di virus itu ya, itu kan lysine residue itu kan secara konsensus itu kan dia berperan pada replikasi virus ya. Salah satu yang paling mudah menggantinya adalah license itu list menjadi RC-GERID RC. Salah satunya itu. Makanya kalau kita lihat paper-paper tentang replikasi virus, mereka akan biasanya mengganti lysine menjadi RC-GERID. Dan kita bisa lihat walaupun di protein C atau protein atau qPCR nanti akan bagaimana efeknya apa. Tapi kan itu bisa dirubah hanya dengan mengganti satu nukleotida saja. Kemudian untuk kalau misalkan kita membuat double mutant, kita juga harus mempersiapkan bisa langsung kita misalnya mutasinya sebelahan ya, asam amino 100 dan 101. Misalkan kita semuanya akan membuat dua-duanya besar, dua-duanya lysine, karena akan kita ubah jadi RC-GERID semua. Kita salah satu caranya adalah kita desain primernya mau langsung jadi dua RC-GERID atau satu RC-GERID dulu. Kemudian setelah jadi satu mutan, kita bikin mutan sebelahnya dengan mendesain primer yang baru lagi. Jangan sampai kita membuat dua primer, misalkan primer kan misalkan kita mau misalkan ada amino asid 101, 102. Itu misalkan ya lysine, kemudian kita rubah jadi RC-GERID. Kita mau lihat nih salah satunya. Nah, 101 kalau jadi RC-GERID efeknya apa? 102 kalau jadi RC-GERID efeknya apa? Pasti kan beda. Bisa jadi beda, bisa jadi tidak. Ternyata efeknya berbeda. Tapi kalau kita rubah dua-duanya gimana nih? Ternyata efeknya jauh lebih berbeda lagi. Tapi ketika kita cuma bikin dua primer karena 101 dan 101R atau 102R itu misalnya RC-GERID kan, RC-GERID. Kita nggak bisa nih buat double mutan dari dua primer itu. Jadi misalkan dari mutan 101 kita mau jadiin tambahin mutan 102, kan tinggal pakai primer 102 ini. Itu akan jadi mismatch. Karena yang mutan 102 kan masih batat ini, yang satu-satunya. Nah, jadi kita harus mendesain primer. Kalau kita intention kita untuk mendapatkan double mutan, triple mutan, 4 mutan, tapi kalau dia jarak mutannya berbeda itu lebih enak sebenarnya. Tapi tetap harus pilihannya bikin 1-1-1-1, kemudian baru kita mendapatkan double mutan, triple mutan, quadruple mutan. Terus kedua adalah TM value-nya. Jadi kita akan hitung, nanti kan ada caranya. Kalau GC itu kan nanti kali 4, kalau TA itu kali 2. Jadi nanti kita jumlahkan GC-nya berapa, TA-nya berapa, kira-kira 50 atau 60, atau kadang 53 derajat atau 56 derajat, atau berapapun yang kita dapatkan angkanya itu. Atau kalau biasanya dari company sudah disediakan sih, oh ini TM value-nya segini, gitu kan. Kita bisa berharap dengan TM value segitu, kita bisa optimal kondisinya. Kemudian ya itu satu lagi yang template-nya berapa, kemudian DPN1-nya itu juga penting. Dalam DPN1 itu kadang inkubasi 1 jam itu cukup untuk beberapa mutan, tapi kadang tidak cukup, kadang 3 jam cukup, kadang 3 jam pun nggak cukup, kadang overnight ya bisa 12 jam, 14 jam. Kadang kalau kita cuma tambahkan misalkan 2 mikro itu nggak cukup, bisa butuh 3 atau bahkan 1 aja udah cukup, 1,5, 2, mungkin kita bisa itulah seninya di situ kita harus karena memang sih. Tapi kalau kita bisa sequence sendiri kan tentunya kita lebih menyirit biaya ya dibandingkan kita nggak bisa sequence, karena sekali sequence kan bisa kita habis berapa juta sendiri hanya untuk sequence, dan kan belum tentu semuanya jadi mutan. Kadang kan 20% kemungkinan kok begitu. Kalau dari tipsnya sih itu sih. Kalau kita sih biasanya saatnya sih template-nya, kemudian atau cycle-nya misalkan dari 10 kita jadikan 15, atau kita jadikan 20 bisa. Tapi ketika kita 15 cycle-nya, oh ternyata kolonnya nggak banyak nih, tapi ketika 20 kolonnya cukup. Dan kemudian kita pilih pemilihan koloni juga penting. Jadi kita lihat koloninya. Mungkin ada beberapa kalau senior kami itu ada beberapa yang bilang kamu cari koloni yang paling bentuknya paling gede bagus, bukan yang kontam ya, maksudnya benar-benar simpel koloni bagus kan, lebih bagus lagi di pinggir. kita bisa juga melakukan itu karena ya memang belum ada pembuktian, tapi dari experience dari senior kita jadi tahu karena hal-hal ini kan seperti ini nggak ada di buku ya. Gimana caranya tips memilih pola yang baik, gimana caranya optimasi itu semua kan case by case, jadi tergantung plasmidnya, tergantung lokasinya, tergantung semuanya. Tapi dengan kita bisa mendapatkan mutasi itu dengan sequence yang baik, tentunya kita akan bisa mendapatkan hasil yang lebih baik. Mungkin
because of the time limitation, I think we will close this discussion session. After this, you can do the process. It may be only 2 or 3 minutes, and after that, we will close this session. Thank you. Okay, everyone, I will close this session. Thank you so much, Dr. Gandhi, for the very helpful lecture. And thank you so much for coming. And I hope the lecture that delivered by Dr. Gandhi can enhance our knowledge about PCR mutagenesis at Gandi. Thank you everyone for your coming. Good afternoon. Assalamualaikum warahmatullahi wabarakatuh. And for the Zoom participants, you can fill the attendance. The link for attendance will be available in the chat box. Thank you. yang di sini habis ini boleh ke lantai 1 ya untuk foto bersama.
Oke, terima kasih.